Hi, how are you? I hope you're doing great. So, today I'm going to explain the carbohydrate metabolism. But before that, you have to master the first video that I recently posted. So, I will be explaining you glycolysis, link reaction, Krebs cycle, electron transport chain, glycogenesis, glycogenolysis, and finally the pentose phosphate pathway. In addition, there is a small portion of about 2 to 3 minutes of about galactose metabolism and fructose metabolism. I want to explain just one thing, that this biochemistry thing is really very easy if you understand the basic concepts. Initially, for the 5 to 10 minutes, it will be a little heavy, but once you understand that, for the rest of the time you will start to enjoy all that information. All right, so let's start today. Thank you. So before I proceed to the glycolysis and link reaction and Krebs cycle, I just want to recap the few basic concepts. The ultimate source of energy for this planet Earth is sunlight, and that sunlight is trapped by the plants in the process called photosynthesis. And in photosynthesis, this carbon dioxide is reduced to food. It is reduced to food here, glucose, and water is oxidized to oxygen. Right? This is photosynthesis, and the reverse of this is called respiration, that happens in animals and human beings. And see that the food is oxidized. The food is oxidized to carbon dioxide, and oxygen is reduced to water. All right, and in that process we get about 36 to 38 ATP molecules upon the oxidation of glucose. Here, oxidation is actually the loss of electrons, while reduction is the gain of electrons. And you can remember this thing: oil rig. Oxidation is loss of electrons, while reduction is gain of electrons. So more reduced is the substance. Upon oxidation, it produces more energy. I mean, if there is more reduction of the substance, for example, if we see that palmitic acid has a lot of hydrogen but only two oxygen atoms. All right, and so upon oxidation, palmitic acid produces about 129 ATP, while in case of glucose, which is less reduced compared to palmitic acid because it has a lot of oxygens here, right, it produces about 38 ATP compared to that of palmitic acid, produces 129 ATP. And as I told earlier, if something is oxidized, if something is oxidized, I mean it loses electrons, then some other thing is reduced that gains electrons. So why is it important? This is important to understand the whole energy metabolism. Food is oxidized to carbon dioxide, and when food is oxidized to carbon dioxide, some other thing has to be reduced. And what is that some other thing? That is NAD and FAD, which are actually the coenzymes. NAD is derived from niacin, while FAD is derived from riboflavin. Right? These are the vitamins, water-soluble vitamins. So NAD and FAD are reduced to NADH and FADH2. And in all those pathways, for example, glycolysis, link reaction, Krebs cycle, fructose and galactose oxidation, fatty acid oxidation, amino acid oxidation, in summary, all food oxidation. All that food oxidation produces NADH. I mean, all that food oxidation reduces NAD to NADH. And now this NADH has to be oxidized back to NAD, and once this is oxidized back to NAD, it has to lose electrons, because oxidation is loss of electrons. So it will lose electrons, and those electrons will be gained by oxygen, and oxygen will be reduced to water. And once this oxidation happens here in electron transport chain, oxidation of NADH and FADH happens in electron transport chain, oxygen is reduced to water, we get ATP. And this is the ultimate source of energy production in the cell, and that is electron transport chain. All right, so this is all the story. I mean, if you see that food is oxidized to carbon dioxide, NAD and FAD are reduced to NADH and FADH2, and when NADH and FADH2 are oxidized back to NAD and FAD, they reduce oxygen to water, and in that process we get ATP during electron transport chain. So, so now we see how those concepts relate in the real cell. You say that in the cell there is cytoplasm, and the major pathway that is happening in the cytoplasm is glycolysis. And glycolysis, as I told earlier, it's oxidation of glucose to pyruvate. Oxidation of glucose to pyruvate is called glycolysis. You see that glucose has 6 carbons while pyruvate has 3 carbons. So actually one glucose molecule produces 2 pyruvate. So we get 2 pyruvate when glucose is oxidized to pyruvate. And in that process, NAD is reduced to NADH. 2 NAD are reduced to 2 NADH, while 2 ADP are phosphorylated to 2 ATP. So the net product of glycolysis is 2 NADH, 2 ATP, and 2 pyruvate. We will study that in a while. And then this pyruvate enters into the mitochondria. And in the mitochondria, this pyruvate is oxidized to acetyl-CoA. So 2 pyruvate are entering into the mitochondria, so 2 acetyl-CoA will be produced in the mitochondria from one glucose molecule. So 2 pyruvate when are oxidized to acetyl-CoA, so we get 2 carbon dioxide, and similarly we get 2 NADH from 2 NAD. Here I want to emphasize, you see here are 3 carbons. Pyruvate has 3 carbons, whereas acetyl-CoA has 2 carbons. So 1 carbon actually goes to carbon dioxide. So as there are 2 pyruvates entering in here, so we'll get 2 carbon dioxide from 2 pyruvate, and definitely we will get 2 acetyl-CoA. And the net product of link reaction is 2 NADH. Alright, so clear here from up till now. So move forward. Now this acetyl-CoA enters into Krebs cycle, and once this enters into the Krebs cycle, we get from these 2 acetyl-CoA, we get 4 carbon dioxide. So all this acetyl-CoA actually goes to carbon dioxide, because 2 acetyl-CoA will produce 4 carbon dioxide. 1 acetyl-CoA will produce 2 carbon dioxide. And once this oxidation happens in Krebs cycle, we get 4 NADH, 2 FADH2, and 2 GTP. So this is the summary of Krebs cycle. So like the whole product of Krebs cycle is 6 NADH, 2 FADH2, and 2 GTP. And note here, this we are talking about 2 acetyl-CoA. When 2 acetyl-CoA enters into Krebs cycle, we get 6 NADH, 2 FADH2, and 2 GTP. And now you can calculate what will happen if 1 acetyl-CoA is oxidized in Krebs cycle. Like we'll get 3 NADH, 1 FADH2, and 1 GTP. So this is how the glucose first is oxidized to pyruvate, that pyruvate is oxidized to acetyl-CoA, and then acetyl-CoA enters into the Krebs cycle to make carbon dioxide. And in whole that process, what did we get? We got 10 NADH, we got 2 FADH2, and we got 2 ATP, these ATP from glycolysis, and we got 2 GTP from Krebs cycle. So total we got this product. And here I just want to emphasize one thing, that when 1 NADH is oxidized in electron transport chain, it produces 3 ATP, approximately 3 ATP, while when 1 FADH2 is oxidized in electron transport chain, it produces 2 ATP. So we got 2 FADH, 2 when these are oxidized to 2 FAD, we get 4 ATP, and 10 NADH, so 10 NADH will be going to produce 30 ATP. 2 ATP from glycolysis, 2 GTP from Krebs cycle, so the total we got 38 ATP. So this is the whole summary of the energy metabolism. I hope this is clear. Please spend some time over this video and this will be clear. So let's start glycolysis. You know that glycolysis is the oxidation of glucose to pyruvate. First of all, glucose enters into the cell. Glucose enters into the cell via transporter, which is called glucose transporter or GLUT. There are 4 types of GLUT: GLUT 1, 2, 3, and 4. GLUT 1 and 3 are present in most tissues, brain, and red blood cells, and they mediate the basal uptake of glucose. While GLUT 2 is present in liver and pancreas, while GLUT 4 is present in muscles, skeletal muscles, and adipose tissue. So glucose enters into the cell, and the glucose is a naughty molecule. It doesn't remain in the cell. As soon as it gets into the cell, it leaks out of the cell. So we need to trap this glucose. So glucose is trapped by an enzyme which is called hexokinase, right? And it is converted into glucose 6-phosphate. Here an important thing, whenever the word kinase comes, ATP is either converted into ADP or ADP is converted into ATP. So whenever there is phosphorylation or dephosphorylation of ATP into ADP, so the enzyme is kinase. Then glucose 6-phosphate makes fructose 6-phosphate by an isomerase. A fructose 6-phosphate makes fructose 1,6-bisphosphate that is cleaved to make glyceraldehyde and dihydroxyacetone. Glyceraldehyde 3-phosphate then makes 1,3-bisphosphoglycerate, and that makes 3-phosphoglycerate, and then 2-phosphoglycerate, and then 2-phosphoenol pyruvate, and finally the pyruvate. So this is the whole story. But you are not supposed to worry about all this pathway, and it's not difficult. It's not relevant to learn all this pathway. So what is relevant is that you see all the reactions of glycolysis are reversible, except the three reactions. And what are those three reactions? First one is this. The first step, when glucose is converted to glucose 1-phosphate. This is the first step that is irreversible. And the second step, when fructose 6-phosphate makes fructose 1,6-bisphosphate. This is the second step, which is irreversible. And the last step, when phosphoenol pyruvate is converted into pyruvate, that is the third irreversible step. And if we could reverse these three irreversible steps, we can get glucose from pyruvate. And this is what actually happens in gluconeogenesis. In gluconeogenesis, we get glucose from pyruvate. This is an important point. You have to remember that there are three irreversible steps in glycolysis, and if we could reverse those three steps, we can get glucose back from pyruvate. Alright. Another important thing to remember is that, see that here ATP is converting into ATP, we get kinase. And here again, ATP is broken down to ADP, and again the enzyme is kinase, which is phosphofructokinase, and this is the rate limiting step of glycolysis, PFK1. It's not GFK1, John F. Kennedy, it is PFK1. So PFK1 is the rate limiting step for the glycolysis, and this is indirectly activated by insulin. This is indirectly activated by insulin because fructose 6-phosphate first makes fructose 2,6-bisphosphate, and then fructose 2,6-bisphosphate activates PFK1. So actually insulin is responsible for activating this step. And another important thing is that whenever the word dehydrogenase comes, it means NAD is going to be reduced to NADH, or FAD is going to be reduced to FADH. So there is dehydrogenase. So you see that here kinase, hexokinase, phosphofructokinase, and now again you see that here again ADP is converting into ATP. The enzyme is phosphoglycerate kinase, and in the last step pyruvate kinase, and again ADP is converted into ATP. Whenever the word kinase comes, it means ADP is converting into ATP, or ATP is converting into ADP. And whenever the dehydrogenase comes, this means it is going to remove hydrogen from the substrate. It is going to remove hydrogen from the substrate, and those hydrogens are going to NAD or FAD to make NADH and FADH, right? So this is an important step. As I told earlier, that the net result of glycolysis is that we get 2 NADH and 2 ATP. It's a little tricky, but really very easy. You have to just know that glucose has 6 carbons. Glucose has 6 carbons, and from 6 carbon glucose we get 6 carbon fructose. But once the 6 carbon fructose is broken down to glyceraldehyde and dihydroxyacetone phosphate, we get 3 carbon glyceraldehyde and 3 carbon trihydroxyacetone phosphate. You remember from my summary that these are the trioses. And this dihydroxyacetone phosphate again makes glyceraldehyde. So in effect, we are getting 2 glyceraldehydes from 1 glucose. And once these 2 glyceraldehydes are processed in glycolysis, we get 2 NADH. And we get 2 ATP here. And we get 2 ATP here. So actually there are 4 ATP. But in the initial process, these 2 ATPs already used. Here, number one at one place, you see that ATP is used in here, and the second ATP is used in here. So in effect, we get 4 minus 2, that is equal to 2 ATPs, right? And we get 2 NADH. And now, another important point is that this NADH, which is being produced here, these two NADH which are being produced here, they have to be oxidized back to NAD, and this happens in mitochondria during electron transport chain in the presence of oxygen. But if there is no mitochondria or there is no oxygen, for example, there will be no oxygen in case of hypoxia or anaerobic respiration, and there will be no mitochondria in case of red blood cells. But in that condition again we have to oxidize these NADH back to NAD. And this happens when, and this is anaerobic. In anaerobic conditions, number one, you have to remember two things. There is no oxygen or there is no mitochondria. There is no mitochondria in case of red blood cells, and there is no oxygen in case of sternus oxidase or anaerobic conditions. In that condition, NADH is oxidized back to NAD. NADH is oxidized back to NAD by the conversion of pyruvate to lactate. By the conversion of pyruvate to lactate. That is the reason pyruvate is converted into lactate, to oxidize this NADH back to NAD in the absence of mitochondria or oxygen. And this lactate, as I explained in my first video, this lactate goes to the liver where it is converted back to pyruvate in Cori cycle, right? So in summary, the pyruvate we got from glycolysis. In addition, we got 2 NADH and 2 ATP. And here this pyruvate enters into the mitochondria, and pyruvate is converted into acetyl-CoA. The reaction is called link reaction, and pyruvate dehydrogenase. Again, the enzyme is dehydrogenase, so it means NAD has to be reduced to NADH. We see in a while. And then this acetyl-CoA goes to Krebs cycle, in the TCA cycle, or it can be used for fatty acid synthesis. We'll see in a while. All right. And the last thing to point in the glycolysis is that dihydroxyacetone phosphate that is produced during glycolysis, this is important to produce glycerol 3-phosphate. Again, the enzyme is dehydrogenase, so NAD has to be reduced to NADH. And this glycerol 3-phosphate is important in triglyceride synthesis and fat synthesis. Glycerol plus fatty acids make fat or triglyceride. And this glycerol 3-phosphate is also important for electron shuttle. Why this electron shuttle? This electron shuttle is important because these NADH are produced in cytoplasm. So the electrons of NADH cannot go directly to the mitochondria. So they need a shuttle, and that shuttle is provided by glycerol 3-phosphate. So these are the important things in glycolysis. Again, I revise, there are 3 irreversible steps that need to be reversed to make glucose again from pyruvate, and that makes gluconeogenesis. Another important point, the pyruvate under anaerobic conditions makes lactate, and whenever the enzyme kinase comes, ATP is going to be phosphorylated to ADP is going to be phosphorylated to ATP, or ATP to ADP. And whenever the enzyme dehydrogenase comes, it means NAD is going to be reduced to NADH and FAD to FADH2. So now we move forward to link reaction. We got pyruvate from glucose during glycolysis, and now these pyruvate enters into the mitochondria. So these reactions are actually happening in mitochondria. These reactions are happening in mitochondria. Pyruvate is oxidized to acetyl-CoA. You have to remember that pyruvate contains 3 carbon while acetyl-CoA has 2 carbons. So the 1 carbon is used in making carbon dioxide. Right? The enzyme is dehydrogenase, so NAD must have to be reduced to NADH, and this is link reaction. Now the acetyl-CoA, which is produced in the link reaction, it goes to citric acid cycle, which is also called tricarboxylic acid cycle or Krebs cycle. And this acetyl-CoA can also be used to make fatty acids. We will see how. Now a few words about galactose metabolism. So we get galactose in our diet through lactose. Lactose is hydrolyzed or digested by an enzyme lactase to make glucose and galactose. You know, glucose is going to make pyruvate. Now this galactose is absorbed into the blood, and from the blood it goes to the cells. Again, this first is trapped by the enzyme galactokinase, and we get galactose 1-phosphate. Then that galactose 1-phosphate makes glucose 1-phosphate, and then this glucose 1-phosphate makes glucose 6-phosphate, and then this glucose phosphate move into the glycolysis to make pyruvate. So actually galactose also makes pyruvate by this process. And just an important point, and this is really very important, very important, that if there is high level of galactose in the blood because of galactokinase deficiency or by galactose 1-phosphate uridyl transferase deficiency, galactose accumulates in the blood, and that is called galactosemia. And if there is high level of galactose in the blood, this galactose enters into the lens, and inside the lens there is an enzyme which is called aldose reductase, and that aldose reductase reduce this galactose to galactitol, and this galactitol exerts osmotic pressure, and because of that osmotic pressure there is swelling and cataracts in the patient who has this galactosemia. And this actually is also the basis of retinopathy and diabetes. If we have high level of glucose in the blood, if we have high level of glucose in the blood, that glucose enters into the lens, and the same enzyme aldose reductase, because glucose is also an aldose like galactose, aldose reductase converts sorbitol glucose into sorbitol, and sorbitol exerts osmotic pressure, and because of that osmotic pressure there is swelling in the lens and the cataract formation. So that is the basis of retinopathy and diabetics. So now few words about fructose metabolism. And in the fructose, again fructose, we get fructose from sucrose, and sucrose is hydrolyzed to make glucose and fructose. Glucose follows the glycolysis, and fructose again is first phosphorylated to fructose 1-phosphate, and then this fructose 1-phosphate, here the enzyme is aldolase B instead of aldolase A, that is used in glycolysis. This fructose 1-phosphate is cleaved to dihydroxyacetone phosphate and glyceraldehyde. This dihydroxyacetone phosphate then can go to the glycolysis or glycogenesis and gluconeogenesis, we will see. And this glyceraldehyde can be converted to glyceraldehyde 3-phosphate, and then this glyceraldehyde 3-phosphate makes glycolysis. So in effect, either this is galactose or this is fructose, they actually join in glycolysis to make pyruvate. Alright. Well, we now move forward to Krebs cycle. We now know that we got pyruvate from glucose, also we get pyruvate from amino acids and from galactose and fructose. This pyruvate in-link reaction makes acetyl-CoA. Acetyl-CoA in addition also comes by the oxidation of ketones, fatty acids and alcohol. Now this acetyl-CoA enters into the Krebs cycle, which is the most important cycle, most important pathway in entire metabolism. Now this acetyl-CoA makes citrate, then isocitrate, alpha-ketoglutarate, then succinyl-CoA, succinate, fumarate, malate, oxaloacetate, and then the citrate. This keeps on repeating. And here the important thing is that dehydrogenases. And now you know what are dehydrogenases. Dehydrogenases, whenever the word dehydrogenase comes, NAD has to be reduced to NADH. So this is the first NADH we get in the Krebs cycle. This is the second NADH, so there will be dehydrogenase. Yes, there is dehydrogenase, alpha ketoglutarate dehydrogenase, and then there is another FADH2. There should be a dehydrogenase, and the dehydrogenase is succinate dehydrogenase, and then malate dehydrogenase. So there should be NADH, and this is the third NADH. First NADH, second NADH, and third NADH. So actually we get 3 NADH, one molecule of acetyl-CoA enters into the Krebs cycle. In addition, we get 1 FADH2. Okay. Now you see that there is one carbon dioxide here and the second carbon dioxide here. So this is the acetyl-CoA carbon dioxide. This is the carbons of acetyl-CoA. The first carbon dioxide and the second carbon dioxide. So the rate limiting step for this pathway is isocitrate dehydrogenase. If isocitrate dehydrogenase is active, is fast, the cycle will be fast, and if it is slow, the cycle will be slow. So let's see which things make it fast. If there is less NADH, if there is less NADH in the cell, if there is less FADH2 in the cell, and if there is less ATP in the cells, isocitrate dehydrogenase will be fast. And if there is more AMP in the cell, the isocitrate dehydrogenase will be fast. But if these things are high, if NADH, FADH2, ATP and ATP are high while AMP is low, and NADH, FADH2 and ATP are high, then isocitrate dehydrogenase slows down. And once this slows down, citrate starts to accumulate. And if citrate starts to accumulate, this citrate leaves the mitochondria and enters into the cytoplasm for fatty acid synthesis and cholesterol synthesis. And cholesterol synthesis via the citrate shutdown. This is an important point. If isocitrate dehydrogenase is slow because of excess of NADH, FADH2 and ATP, then the citrate starts to accumulate and that citrate leaves the cell to make fatty acid and cholesterol, right? And if you remember from the overview of energy metabolism, the first lecture, all that acetyl CoA starts making fatty acid and cholesterol when the energy needs of the cells are met. All right. Another important point is that this succinyl CoA, which is produced in Krebs cycle, this is important for making heme. Heme is not only important for hemoglobin and myoglobin, but it is also important for making cytochromes. And cytochromes are very much important in cytochromes are very much important in electron transport chain as well as in the liver metabolism. In addition, heme is also an important component of catalase and peroxidase, which are antioxidants, which acts as antioxidants, catalase and peroxidase. Right? This is an important point. Succinyl CoA makes heme, and heme is not only important in hemoglobin and myoglobin, it also is important for cytochromes and catalase and peroxidase. And then fumarate. You see that this fumarate, this can link with urea cycle. Urea cycle produces fumarate, and then this fumarate becomes the part of the Krebs cycle. Right? Another important point is that this malate leaves the cell and is important in gluconeogenesis. We will see that in a while. These are the important points. You see that this is actually the Krebs cycle. Krebs cycle is important for fatty acid synthesis. When the Krebs cycle is slow, the fatty acid and cholesterol synthesis happens from citrate, and also there is a heme formation from succinyl CoA in the Krebs cycle. So now we see the electron transport chain. It is really very easy. You see that all those pathways like link reaction, like Krebs cycle, like beta oxidation of fatty acids, or the amino acid oxidation, or the ketone body oxidation, or alcohol oxidation, they produced NADH. Now these NADH gives their electrons to electron transport chain, This is taken by NADH dehydrogenase complex 1, and now these electrons are handed over to coenzyme Q. Now coenzyme Q also gets electron from glycerol phosphate shuttle, and if you remember, glycerol phosphate was produced in glycolysis and was important in transferring the electrons from glycolysis. So this glycerol phosphate shuttle, FADH2, gives its electron to coenzyme Q. Similarly, succinate dehydrogenase complex 2, which is also the part of the Krebs cycle, gives its electron to coenzyme Q, and then fatty acid oxidation produces FADH2, and those FADH2 are taken up by the coenzyme Q. Now the coenzyme Q hands over their electrons to cytochrome B by C1 or complex 3, and complex 3 hands over electrons to cytochrome C, and cytochrome C hands over the electron to cytochrome A by A3 or the complex 4, and finally these electrons are accepted by oxygen, they are gained by oxygen, and oxygen is reduced to water. And now, and you know that electrons move, current is produced, electricity is produced, energy is produced. That energy is sufficient to push these protons from the matrix into the intermembrane space. You see that this inner membrane, this is the intermembrane space, and now in the intermembrane space there are a lot of protons accumulated because of the movement of electrons in the electron transport chain. I again say that when electrons are transported in the electron transport chain, current is produced. That energy is sufficient to push the protons from the mitochondria into the intermembrane space. And once these protons are accumulated over here, they move back to mitochondrial matrix. By F0 F1 complex. And when they move back to the mitochondrial matrix, again the charges are moving, so the energy is produced. That energy is sufficient to convert this ADP into ATP, and then that ATP moves out of the cell by ATP-ADP translocase, and it is used in different energy purposes. All right, so this is the whole story, right? So if I make it a little zoom in, right, so you see that so this is the whole story. Electrons from NADH and FADH2 are handed over to the electron transport chain. In the electron transport chain, these electrons move And when these electrons move in the electron transport chain, energy is produced. That energy pushes the protons from the mitochondrial matrix into the intermembrane space, and when these protons move back to the mitochondrial matrix, energy is produced, and that energy converts ATP into ATP, and that ATP is used in different cell processes. But sometimes, because of the presence of uncoupler proteins, which are aspirin in high doses thermogenin, these protons do not couple with F0 F1 complex. Rather they move directly from the intermembrane space back to the mitochondrial matrix, and in that process energy is produced, and that energy is lost in the form of heat instead of ATP. Another important point, the complex 4 of the electron transport chain is inhibited by cyanide and carbon monoxide. Complex 5, it's also called complex 5, F0 F1 complex, is inhibited by oligomycin, while complex 1 is inhibited by barbiturates and rotenone. While rotenone is an insecticide, and barbiturates, they are used to depress the central nervous system. All right, and they are also used as anesthetics, as an adjuncts in anesthetics. So this is electron transport chain. All right. So now we move to glycogenesis. We know that this glucose is entering into the liver. All right, that glucose makes glucose 6-phosphate first in the glycolysis. Right, now from the glucose 6-phosphate, it starts making glucose 1-phosphate, and then that glucose 1-phosphate makes UDP glucose, and then that UDP glucose by the enzyme glycogen synthase and branching enzyme makes glycogen. So this was one step. The glucose 6-phosphate starts making glucose 1-phosphate, and then glucose 1-phosphate through UDP glucose starts making glycogen. And glycogen synthesis happens in the presence of insulin. All synthesis happens under the influence of insulin. Right? And insulin actually activates glycogen synthase, and glycogen synthase starts making glycogen. This glycogen is stored in the liver. But in the glycogenolysis, when we need glucose during fasting or starving, there are the hormones like epinephrine, like glucagon. They now activate glycogen phosphorylase and debranching enzyme. Glycogen phosphorylase, and similarly AMP in the muscles. When there is less energy in the AMP is actually the indicator of the energy. I mean, if there is less energy, there will be more AMP, and if there is more energy, there will be a lot of ATP. So now this AMP or the epinephrine or glucagon and even cortisol as well, right? So these hormones activate glycogen phosphorylase and debranching enzyme, and glycogen is broken down to glucose 1-phosphate. And now this glucose 1-phosphates make glucose 6-phosphate, and that glucose 6-phosphate can be used in glycolysis to make pyruvate. But in the starvation, we need glucose in the blood. So this glucose 6-phosphate makes glucose, and this was the irreversible step of the glycolysis, which has now been reversed by the enzyme glucose 6-phosphatase, and this is an important enzyme of gluconeogenesis, actually the last enzyme of the gluconeogenesis we will see. So glucose 6-phosphatase actually converts glucose 6-phosphate back to glucose, and now glucose is free to leak out of the cell and enter into the blood. Right, so glycogen breakdown produces glucose, and that glucose increases glucose level. Well, this concludes major portion of carbohydrate metabolism. Only two topics are left. One is gluconeogenesis, and the other one is hexose monophosphate shunt or the pentose phosphate pathway. I will be making a video on those two topics soon. Please leave your feedback, and if you have any question regarding this video, you can post them in comment. Thank you so much. Hope to see you soon. Thank you. Bye bye.